Hello guys, welcome back to my channel. Today we will see about the mechanism of replicative transposition. First of all, we know that transposable elements are nothing but the jumping genes or moving DNA elements which can move from one specific donor position to recipient. Okay. The mechanism of transposition can be done via two different mechanisms, that is replicative transposition and the second one is non-replicative transposition. Okay, so replicative transposition is also known as copy paste transposition and non-replicative transposition is known as cut and paste mechanism or cut and paste transposition. So in this video we will see about the replicative transposition mechanism. Through the diagrammatic representation I will explain you about the transposition, that is replicative transposition. It is also known as copy paste mechanism. because why replicative means क्या होगा, जो भी ट्रांसपोजेबल एलिमेंट होगा डोनर डीएनए में वो रेप्लिकेट होगा. दैट मीन्स टू डुप्लीकेट कॉपीज विल बी फॉर्म. that means the original copy will remain at the donor site also, and also it will generate one new replicative copy of transposable element in recipient DNA also. Okay, so two different replicas or replicons will be formed, thus it is known as replicative transposition. So here in this diagram the first step will be what, in the donor DNA, wherever the transposable element is present at the insertional site sequence IRS, which is showing in the diagram, here transposase enzyme will come and bind to this. Okay, so first of all what will happen, transposase enzyme will bind at the insertional sequences, that means which the sequences which are present at the ends of the transposable elements will bind, transposase will bind at this sequence. Okay, it can bind to target site sequence also. Then the nick will be created at the 3' end of the DNA element. Okay, transposable element is obviously double stranded DNA element, so it will have 3' OH and 5' phosphate element. Okay, so it will create nick at the 3' OH. Okay, then the other single stranded cuts will be occurred. Okay, at the 3' OH in donor DNA, at the 3' OH transposase enzyme will bind and will cut at the 3' OH DNA and will create a nick. And at the specific target site sequence also transposase enzyme can cause a nick at the 5' OH. Okay, then the 3' ends of the transposons are joined to 5' ends of the target DNA. So it will happen in the crossover mechanism, where 3' OH of the cut transposable element will bind to the 5' ends of the target DNA, which will be present in the recipient DNA. Okay, so crossover structure will be formed. Then the DNA synthesis fills in single stranded regions. Then the DNA polymerase enzyme will bind to the 3' OH and will continue the DNA formation. Okay, so DNA polymerase enzyme जो भी होगा वो क्या करेगा, DNA synthesis करेगा, DNA strand preparation start करेगा, will bind to the 3' OH. Okay, then the co-integrate structure will be formed. Co-integrate structure मतलब क्या, it is the fusion of donor DNA and recipient DNA, where it will have two new DNA molecule, two new strands of the single stranded molecule of donor DNA as well as recipient DNA. Thus it is known as co-integrate, because it has two new copies of you can say transposable element. Okay, then at the last step resolution will occur, where recombination takes place. Okay, why resolvase enzyme will resolve the donor DNA as well as recipient DNA, and thus creating two different duplicated copies of transposable elements having two DNAs each with transposable element. Okay, so in this way replicative transposition happens.